Hi, my name is Kaylee, and I'm going to be explaining what my research is. So, in scientific terms, my research is trying to discover new small molecule inhibitors of dealanine ligase and Staphylococcus aureus using high throughput virtual screening. But what does all of that really mean? But what does all of that really mean, and why is it important? In the next few minutes, I'm going to break down this sentence into digestible terms and explain what my research is all about. Let's start with Staphylococcus aureus. S. aureus is the bacteria that causes staph infections and is the leading cause of infections in U.S. healthcare facilities. Exposure to the S. aureus pathogen can lead to a variety of ailments, including cellulitis, pneumonia, endocarditis, toxic shock syndrome, bacteremia, and sepsis. And I'm sure you're thinking, don't we have drugs to treat that? And my answer is yes and no. Antibiotic resistance is a growing problem for a multitude of bacterial infections, including staph. Basically, the bacteria is able to mutate and find alternative ways to function in the presence of these treatments, which is why new treatment methods need to be discovered. The drug development process is very time consuming, and my research is not developing new therapeutics themselves, but it is intended to find new compounds that could be made into new drugs in the future. Now, how do we even do this? Well, I use what's called high throughput virtual screening. On the screen in the top right is a 3D structure of dealanine ligase, or DDL, which is the target protein. This protein is essential in maintaining bacterial cell structure and is the current target of many antibiotics on the market used to treat staph. So we use a software called GOLD, and this can virtually bind compounds against the target protein. Think of the protein like a puzzle with one piece missing and a compound binding to the protein as connecting another puzzle piece. The actual final puzzle piece, or compound that would normally bind to the protein, would allow the protein to function well. If we place a puzzle piece with the right size and shape into the final slot, the puzzle is finished, but hopefully does not create a pretty picture. This research is searching for that final puzzle piece or a compound that will prevent the protein from functioning. Each of these compounds is given a score that tells how well the compound is predicted to bind. In theory, compounds with higher binding scores can bind to the protein well and are considered good candidates to prevent the protein from functioning. The second portion of my research is actually testing the ability of these top-scoring compounds to inhibit the target protein from functioning. We start by growing the target protein from DNA and measure the protein's activity. We then take one of the higher-scoring compounds, bind it to the protein, and measure the protein's activity again. Hopefully, the protein activity is lower, indicating that the compound in question was able to prevent DDL from functioning. This research is not intended to cure staph or to create new drugs, but the compounds that are successful both virtually and in a traditional laboratory setting can potentially be used to create new drug treatments for staph.